This is ARVO 2015, and we're talking to you about the research presentation that you brought to this conference. Tell us a little bit firstly about your background and the laboratory you work in.
Okay. I work at North Carolina State University. I'm a third-year PhD student actively working towards my degree. The lab I am in is under the mentorship of Dr. Chris McGann, and we study the iron metabolism of the eye.
Tell us about the research presentation that you made here.
My research presentation was on hypoxia's effects on the amyloid precursor protein as well as other polarized proteins in the eye, and how the loss of oxygenation to the eye causes these proteins to change their polarized expression. I also undertook looking into how retromer, a protein transport complex, is able to be disrupted as well by hypoxia. Specifically, what we found was that when the eye was exposed to hypoxic conditions, APP changed its polarization. Instead of being released apically, it was released basolaterally out of the retinal pigmented epithelial cells. These cells are the barrier cells of the eye. And when you change the polarization of proteins in these cells, it can dramatically affect the metabolism of the eye. So we saw this dramatic decrease in the basolateral secretion and the apical secretion of APP in hypoxia. We wanted to know what was controlling this secretion besides hypoxia, so we looked into transport complexes. We learned about the retromer complex, which has been shown to transport APP in a polarized fashion in other cells. We then exposed our RPE cells to hypoxia and saw that retromer was also dramatically decreased in its expression. So then I used siRNA to knock down retromer expression, and in all time points in normoxic and hypoxic conditions, we saw significant decreases in APP secretion and expression in our RPE cells. Moving on, we also have discovered that other polarized proteins are affected when hypoxia is introduced into the environment. Specifically, I have found that transferrin receptor, the iron controlling factor in RPE cells, is also downregulated when hypoxia is introduced into the cell. This has been very interesting research, and we hope to continue it further to see how hypoxic conditions can affect other polarized proteins in the eye.
It's very interesting you mentioned transferrin. Dr. Gady Wallstein from University of Pittsburgh has been doing work on hemosiderosis. And apparently excess iron deposits have been shown to accelerate macular degeneration. How do you think this plays into the issue of hypoxia?
Excess iron is widely known to cause an increase in reactive oxygen species in the eye, and when reactive oxygen species are introduced into the ocular environment, this causes widespread disruption of many cellular processes within the eye. So since iron has to be controlled in a very finite way, the cells in the eye need iron to function. If they have too much or too little, these reactive oxygen species can then come into play and disrupt that cellular metabolism.
Tell us about your next steps for this particular line of research.
So my next steps, I'd like to look into other polarized proteins that are also affected by hypoxia and are also likely to be shuttled by retromer in a polarized fashion in the eye. Specifically, I hope to look into the iron transporter DMT1 and see if it is disrupted in the same manner as hypoxia disrupted APP in the transferrin receptor.
Turning to potential translational implications of your work, what do you think your work can contribute to a better understanding of macular degeneration specifically, and what patients might anticipate in the future?
Okay. So APP, which I studied in these hypoxic studies, has been implicated in the onset of macular degeneration. Specifically, APP can be processed into the amyloid beta protein, which is widely implicated also in Alzheimer's disease. But in macular degeneration, it has been found to be present in the fatty deposits that form in the eye called drusen. We believe if we can figure out how hypoxia is affecting the polarized movement of APP, we then may be able to understand how to control this movement and then change the localization of APP from going out of the ocular environment into these drusen deposits and potentially reducing the amount of drusen found in macular degeneration.
Turning back to the professional aspect, what specific research are you intending to conduct as a follow-on to this study?
Specifically, we'd also like to see how, if hypoxia affects its polarized proteins, what other metabolites may it affect in the cell. Since our lab focuses on iron research, we'll follow that vein of research and see how iron is also affected by hypoxic processing of these polarized proteins.
Is there anything else that you'd like to share with us about your work and your laboratory?
Overall, our laboratory has been in existence for over 30 years, and we really have much experience in the iron field as a whole, and we hope to continue this experience. Our next experiments that we are planning in our lab are looking at the glycosylation of iron in hypoxia, which we believe is a field that has been understudied. And so we hope to understand how hypoxia affects the glycosylation of iron regulatory proteins, and then thus how this affects the iron metabolism of the cell overall.
Thank you very, very much, Philip. We appreciate your research and your time.
Thank you.